Sanskrit scholar and researcher in history of Indian medicine. And that's why her main task is to find out the links between oldest Indian literature in medicine and try to relate with modern science to confirm that. So this is a part of that, and I am honoring her request anyway. I need not speak to this sentence. We have just finished an article by Karolinska Institute professor who is editing safety of nanomedicines, and this is, then we have contributed nanomaterials, neurotoxicity, and this will be by Stanford Publishing. That the book is coming in ISI website anyway. So, what is known? There are two ancient system of medicine. One is Chinese, that is a bit older than the Indian ones, and Indian medicine is Ayurveda, and that is written in Sanskrit, and it is translated from the British in more than 200 years ago. Some of the translations are not right, and due to many other causes. And Government of India has published these old documents, 8 million pages digitalized, so we have much more access, and my wife is going through that. So here is, this is for Nagarjuna, the father of Indian pharmacotherapy. And even old rocks, they write some mantras for spiritual things. And now people know that musicology is a subject in neurology, and Dr. Klaus von Wild, whose son was here, and a German scientist, is very much pioneer, treating human diseases by music. But anyway, I don't want to relate those things. And this was the book in 1973 that provides many things about the Indian medicinal concept. So when she researched the old digital documents, we can find the history of nanoparticles is even 5,000 years old. And in these are 4 Vedas called Atharvaveda is medicine. So everything is written about medicine, how you treat, and due to lack of time, I am not telling so many things. But it's everything in a shlok. And this is the first Indian description of Charak Samhita, means the medicine book. And the toxicological sciences is, they have given their own name, and they have divided in 2 parts. Is as you can see, animate and inanimate. Some of the references are given. So the toxicity caused by poisons, minerals, rocks, and so many things that they can be exposed to human beings, they can even lead to death. So I think that is one of the concepts of nanoparticles in early Indian medicine. The rest are, they're talking about venoms. And for the treatment, in Ayurveda, there are different kinds of basmi, these are powders. Powders from different metals, different things, and they mix with different type of chemicals that produce like that. But what are they? They are really nanoparticles. And so that's why it's the Nagarjuna, the name is that. He was the first person of Rashasas, means the neuropharmacology. What are those or powders are used nowadays normally, even now in Indian medicine? They contain gold, silver, copper, lead, tin, iron, mercury, and sulfur. And that's why many Ayurvedic medicines or endogenous medicine from China and India, in many countries, including United States, there is a warning that they should be toxic, and or herbal supplements. So these things are still there. I don't want to go into legality or technicality, but I am telling you only facts. So these powders were used for different kinds of treatment, and as you can see that many of these powders contain different proportions of various so-called nanoparticles nowadays. So there is a need even nowadays, and even people using this Indian medicine for practice, and many diseases are cured. They are still working on the toxicological aspects using modern laboratories. So here, and they also talk about the size. Of course, they measure using very primitive things, and also this to make powders or crucible and other things, but still they are working. So the size was important, and this was discussed earlier. So that is a good idea to test size and age. And we have this Indo-EU project that gives us some opportunity to work. Also, we have Swedish strategic research. This is also they are thinking to make modern medicine combination with previous history of medicine. So here is the group is we have size range, and in this case 20 to 30 nanometer, 50 to 60 nanometer, and 130 to 150 nanometer. We have some idea from the literature and also some of our works that the small size of the nanoparticles can create more neurotoxicity. Then what about age? Age is also an important factor, and therefore 3 different age groups were chosen. I cannot relate age of rats to the human stage of development, but 9 to 10 weeks means they are rather young. Most of you are very pioneer in animal research and have much more knowledge than me. 18 to 20 weeks could be middle, I don't know, human classification, and 30 to 35 weeks, they could be young adult, they could be mature adult, but may not be aged according to our definition. But we have these 3 groups. The doses were very simple, 50 milligram per kilogram. And extension of this work, published in the Society for Neuroscience, that has come in news in neuroscience in nanoparticles, and also after Dosi's work came in the news. So if you are going to Society for Neuroscience, you can see the newspaper and some activity. And of course, when we do with chemistry, we met the Nobel laureate in Italy, discussed something about that, our work, and he is very pleased. And Dr. Stepper is here. He was very kind, allowed us to do a special issue in hot topics in CNS neurological disease and drug targets. This impact factor is also great, and so we are very happy with that. And when we submitted this volume, we submitted to some key scientists there, including Nobel laureates, and this is Dr. Shelley. I will not read, and this is the chemistry Nobel. This is Dr. Ottersen, much more familiar to you. He is now rector of Oslo University, so he left, but he worked as a chief editor of neuroscience for so long time, and well recognized and prestigious neuroscience editors. So we are very happy that we are going in right direction. And again, being a neuropathological laboratory, and she also has interest in neuropathology under Sarvas Navarro. We were in Humboldt Foundation in Germany. So we always use this very breakdown. And the point is, the main point is here, that whether nanoparticle neuroplasticity is associated with nitric oxides and base products, because this is a free radical material. So we try to combine these few things and try to see what is the result. And as you can see that just blood-brain barrier breakdown, 9 to 10 weeks, 18 to 20, and 30 to 35. And this is the distribution. Saline, there is no any change. Copper, 20 to 30 nanometer. The very young, 9 to 10 weeks, and older animals, 30 to 35 weeks, they show much more increase in permeability compared to the middle age, 18 to 20 weeks of rats. And this is quite similar with copper too. And regarding this is the size and this is the age. So different size you can see. So this 20 to 30 nanometer is producing much more damage in case of copper compared to 130 to 150 nanometer. So these are the doses and age. Now this is showing another way Evans blue leakage, and here you can see 20 to 30 nanometer of 3 different ages, and you can see that 18 to 20 weeks are showing less damage compared to the very young and old as compared to this one. 50 to 60 nanometer is lesser, and 130 to 135. So the line is like that, and depending on this, the interesting finding is that no matter what the size of nanoparticles you use, this effect of age is quite similar in all size types. Interestingly though, silver even has a little higher impact, but it may not be statistically significant, but these are the trends. And but the result is that large the size of the nanoparticles, silver, lesser damage to the blood-brain barrier, but the age relation sticks more or less. Aluminum was the least effective, but what is important to us, that even it is least effective, but it has almost similar relation when the age group and the size. So I guess to us this is very important, because the idea is that when many nanomedicine, either in Indian or any other, is given, we should be sensitive, because we can't prescribe the same kind of medicine to very young children and very elderly things for certain kind of things. We should, because the brain stages might be different. So then this was, I have just described the same thing we got with radioiodine compared to Evans blue. Radioiodine is smaller molecular diameter when it mixes in the serum, so it is smaller in size than Evans blue albumin complex. So what we can see, that although there is a little greater extravasation, but the relationship exists. Edema measured as water content. Of course, you can see that with more or less some variations in certain age groups here. The basic mechanisms are very similar. The middle age group is less sensitive as compared to the young and old ones, and also the smaller size of copper and silver are more dangerous, and albumin in general is less. Here we have calculated brain swelling. This is a mathematical expression designed by in 1949 by Elliott and Jasper. This measures the volume changes from the water content in simple measures. 1% increase in brain water, either regional or whole, represents about 4% in volume swelling or expansion. So this is a mathematical conversion to the data, and we are happy to see that even volume swelling calculated in that way also follows the same pattern. Now the North expression. There are two theories that, of course, all kinds of nanoparticles produce oxidative stress, and very little is written about nitric oxide, because this is also a free radical. So we have a large project examining neuronal nitric oxide, immunological nitric oxide that is much more devastating, and endothelial nitric oxide synthase using that. This is also a bit controversial, because if you use inhibitors of nitric oxide synthase, it could also block neuronal and endothelial. So they are very controversial. But here I am showing you some results with neuronal nitric oxide, and as you can see in summary that the relationship still exists in that way, more or less. The small size are more harmful, means increasing more nitric oxide synthase, and aluminum is least effective, and even the age relationship is still there. In some cases here it could be not significant, but more or less the relationship exists. And here is the neuronal damages. There are little differences, but in basic the same pattern is there. When we try to correlate these parameters, and as you can see here that this is the size of nanoparticles and leakage of radioiodine. In case of copper with 3 age group, they are rather highly correlated. This is for aluminum. It is more correlated, tight correlation than aluminum. And copper is also. So I mean, with the size and the changes in leakage, they are tightly correlated. So on this basic observations, I should say still preliminary, neurotoxicity is inversely proportional to the size, and young and elderly are more vulnerable. The question is now that, for example, there are many occasions when in motor vehicle, exposure or on the road, if much more carbon nanoparticles or exhausts are there, young children are packed in car for a long time, and even some elderly are driving. These are the questions that Swedish government is asking to us, and we have no idea. So we want to develop this kind of protocol, and I will show you some histological studies in this case to see what we got. And this is young, adult, and old. These are neuroprotective oxide, copper nanoparticle 50 to 60 nanometer. You can see that young and old, they are showing a bit more as compared to the adult. GFAP, we have shown silver nanoparticles, and young and old, they are apparently from the same areas showing more expression, although this holiness and edema can be seen is quite similar. HNE is staining copper nanoparticles. Here we can see more damage. There's also damage, but I mean it's in the same area, a number of neurons, they appear to be less. Then to prove our hypothesis at light microscopy, we went to electron microscopy because that can give us some better idea. And myelin basic protein, you can see that in young and old, this is almost here, histochemistry is almost damaged, but adult still one can see something. And these are the myelin damage here, beautifully shown by one earlier, because, and you can see here young and old, but in case still we can see there is not that damage as compared to young and old. So neurotoxicity is definitely size related here in our case, and also age related, and probably the basic properties of nanoparticles would also be important. And this is a large study group, and I mention of course government of India, vast countries, Swedish government, including more cooperation, Medical Research Council of India, Swedish Strategic Research, and support from Chinese organization. This is our former Prime Minister from Social Democrat. You also remember he is active supporter of research and development in Sweden. And so this is presentation from Sweden, Uppsala University. Thank you very much. Yes, sir.
Yeah, so the age differences is particularly striking. I mean, these are all post-sexual maturation, and yet their age differences are not terribly great in the way I think a lot of people who do rodent work think. So what do we think could possibly be accounting for these I mean, they're huge differences.
No, there's not huge differences in the age. They are not very
No, but they're huge
old, not very
differences
young.
in
Yeah.
the effects
Yeah.
over a short age range, and
Yeah.
it seems bizarre, actually.
Yeah, to someone it could be, but here we observed and proved in different ways. So we believe that the age differences are real, and of course in terms of the blood-brain barrier maturation, all are matured, and the stress reaction also varies with different age group, and for that reason these age differences are very large. So it depends what one would like to see and how to interpret these data. So for stress reactions and for other kind of treatment and responses, these are much large. We have not studied the other effect of, for example, methamphetamine or something to show this, but only I can say, and these are quite regular findings. So we have to find an explanation. Even the data do not. Yeah, we have to find an explanation, because it can always happen that all our beautiful hypotheses could be destroyed by early facts. So this is the fact. But now we have to find the explanation. If you can't find the explanation, we can't say that these data are predicted here.
It's all the same pattern, that would be much more impressive. I mean, so maybe just be careful with some of the models used.
Well, no, I understand that. What I'm saying is that these are really big effects based upon age, and this is sort of not what I would expect in this sort of an area. Certainly not in that sort of inverted U-shaped effect. And if the effect is real and reproducible, then there's really some very interesting stuff there. find out what's
Might
going
be.
on as
Well,
opposed.
my point was relevant to humans. If you did it in a gerbil or something and it was different, then it's kind of so what. But if you saw the same effect in different species,
Yeah.
Well, yeah, but I
I guess the
understand.
question is, what's the next step? I don't know if you can explain this.
I mean, the relevance to humans beside the point. I mean, what I'm saying is that a lot of people use the rat, and if they don't understand that this is going on, then that really confounds a lot of interpretations of the data that are out there. And so the finding is very intriguing, and it could be very important for the interpretation of a lot of data that's being given.
Yeah, could be, because not only that, what I am afraid to publish some of the data or even present to this august audience is that but I can support some of the things. Barbara Johansson in University of Lund, she did simple experiments, and the paper was published in Actinologica Scandinavica in 1981. And she injected, I think, noradrenaline and adrenaline intravenously and produced hypertension in a short period that disrupts the barrier. And I studied with Yvonne Lee. And what she found, she's a neurologist, if she did the experiments in same age group of same strain of rats in the daytime and in night time, there are huge differences. So one cannot just rule out. And the second point is that eventually I will publish. I'm finding more evidence, then I will tell to scientific community. If the same kind of experiments are done at 3 different seasons, I mean, if there are any psychiatrists here, they can understand that seasonal variation is very, very important. Why some people do suicide in certain part of the year. So I can tell you that we have the experiments, trauma, many other things, done at 3 different time period of the year, not in India, where somebody will say that, well, laboratory temperatures are not maintained. We have proved it in some of the labs in Europe, some of the labs in the United States, and we are getting the same difference. So even we can't think that just doing the same kind of experiments in different seasons will affect results. And therefore, I have seen some laboratories, I am not an expert of data analysis of others, I am only concerned to me, but in some of the laboratories there are wide variations of the data, and some of the factors could not be ruled out. The same other important factor in one laboratory is humidity. In some of the countries like Sweden, for example, in winter, if outside temperature is minus 20 degree or 40 degrees Celsius, Sweden is, they have the resources. So even they maintain humidity at minus 40 in animal labs. So they can say that humidity is clear. But there are many laboratories in so-called developed world, they can't maintain the humidity inside the animal chamber, and the results are different. So the point is that these small differences can make very large difference in the result, and that's why we have variations. I can only have to say this. Yes, please.
These were intraperitoneal injections, is that correct?
Yeah.
Some of these all of these are small enough that they could be taken systemically by inhalation. What would that do to some of these findings? Does it change anything?
We have not been able to develop this inhalation method in our country. And I believe that inhalation experiments can also show some changes, because it depends on how much nanoparticles are inhaled, and there should be individual variation, there should be group variation, but I mean, this is an interesting part, one should do this. In my command, I don't have any laboratory where we can do inhalation.
It would have great clinical application.
Yeah,
if we
that's
could
right.
do that.
That's right. Yes.
Yeah, I wanted to just sort of second Bill's point there. You need all these bits of information, and I'm really glad that you're looking at the trends over an area of size, and of course others could look at it over particle ionization, and others could look at it over degree of plasma and other proteins that are bound to the nanoparticles. All these kinds of fundamental principles about the delivery of nanomaterials is what we need to know, just like we did and learned about chemical delivery some 50 years ago. So we're catching up with this, and I agree with you, Bill, it's very critical. So on top of the idea of the blood-brain barrier transfer distribution once you're inside the animal, Bill's point is that in addition to that, you may have topical application, let's say sunscreens that contain nanomaterials, or you may have inhalation of other kinds of nanomaterials.
Yeah.
We have to learn those principles, add them to what you've generated here, and come up with an overall strategy on how you would evaluate whether or not a compound is going to get to the target. And it's a lot of work, but that's what's going on now. And actually right now, Paul Howard, Roger Buchanan at ASU, many individuals at UALR, UAMS, and the other University of Fayetteville and University of Arkansas Fayetteville, they're all working toward this group search for these kinds of principles. And there's many other labs across the country doing the same thing, but within Arkansas, there is a very systematic approach going on right now to try to get to some of these fundamental principles that you talked about, Bill. And they're really important for the future use of these nanomaterials, which in some ways, some are already available to us, but there's many more that are being developed, and we need to know these basic principles before we can safely use them in the future.
Yeah, that's very important because still we do not know. I think the blood-brain barrier research developed, Paul Ehrlich 1885 or something like that. But still we do not know that whether the same substance is applied inside the lumen and abluminally over the microvessels, they are going to have same effect. Whether the receptor distribution of these biogenic amines or transporters in the luminal side and abluminal side are similar. So that's why we have done some chemical study like serotonin. We have given intravenously, we have applied topically, and the spinal cord and microvessels. I can tell you that if you apply the same amount of serotonin concentration over spinal topic microvessels and brain microvessels, the dose and response is entirely different. So even in the same animal, there are differences in the vascular reactivity in the brain and spinal cord. So these are new things. So we should try to understand, but what we wanted to study in this goal, that there is an effect of age and there is an effect of size. To somebody, it can say there is a dramatic difference between aluminum and copper, and this might be very difficult to explain. But one should try to understand these basic principles and question these things, because that's why the research and congresses are there. We present our data, understand the views, and try to go to the lab and try to investigate from them.